In this video, I'm going to walk you through a de novo assembly of E. coli in SeqMan NGen using Ion Torrent data. So the first step after launching the program is to select your project type, and I'm going to keep the selection of genome assembly and click Next. And then it asks me for my assembly type, and I'm going to select de novo assembly and click Next. Here I need to define my project name, so I'm going to go ahead and type that in. And I also need to specify a project folder for my output files, and I already have a folder set up that I will select. Then I can click Next. And then I'm asked to specify my sequence files, so I'm going to select Ion Torrent from the Read Technology field. And then I can choose the 5 SFF files I have from Ion Torrent that contain my reads. And now that I have those added, I can click Next. And this is the Read Options page. This allows me to adjust the parameters that are used for pre-assembly scans. I'm just going to leave the default settings as they are and click Next. And this page allows me to adjust the assembling parameters for my project. And the only thing I'm going to do here is enter the expected genome length, which is 4.6 million. And I'm going to leave all the other settings as they are because based on the project type, assembly type, and read technology we already selected, SeqMan NGen has already optimized these parameters for me, specifically for a de novo genomic assembly using Ion Torrent data. So although I have the option to adjust any of these parameters, I'm just going to leave them as they are. One additional thing to point out is that on each page of the wizard there is a Help button. And if you click on that button, you will be taken directly to the page in the help that explains each of these options in detail. So if you're ever wondering about changing a parameter and you're not sure what effect it will have, just click on the help button to see a detailed explanation of each option on the page. So I'm going to leave these settings as they are, click Next, and that's all that's necessary for setting up the project. The final step is to click Assemble, and SeqMan NGen will begin assembling our data. Once the assembly is complete, it will automatically be opened in SeqMan Pro for analysis. The first thing you'll see here is the project window, which shows the contigs that were created from the assembly, as well as the length and the number of sequences within each contig. And you can sort this list by clicking on the column headers. I'm going to go ahead and sort by length so that the longest contig is listed at the top. And as you can see, we have some nice large contigs that were created here. I'm going to go ahead and open up contig number 1 by double-clicking on it, and that displays it in the alignment view. The alignment view shows us at the nucleotide level the consensus sequence for the contig, which is shown here at the top, as well as the alignment of all of the sequences making up a contig in the lower section of the window. Now, SeqMan Pro offers several options for annotating your consensus, and I'm going to demonstrate one of those ways, which is to collect features from a BLAST search. So to do that, I'm going to select the entire consensus by going to Edit, Select All, and then I'm going to choose Net Search, BLAST Selection, and click OK. Once the search is complete, you can select from the results the sequence that you want to get the features from. And it looks like several of these at the top have the same % query coverage and score. So I'm just going to select the top result and then click on the Collect Features button. SeqMan will prompt me to save that sequence, so I will click OK. And then what's happening is that SeqMan is taking all of the features from within the matching portion of that alignment and adding them to my consensus sequence. And once they've been added, they will be displayed in the feature table, which is what we're looking at here. Now to see a graphical representation of this contig, I can go to Contig, Strategy View, and from here you can see the depth of coverage for the contig represented in the depth of coverage graph. And you can also see all of the annotations that we just added shown in the Features section. And hovering over any of these annotations will give you details for that specific feature. If you have further questions about assembling or analyzing Ion Torrent data using SeqMan NGen, please visit our website at DNASTAR.com, or contact us at support@dnastar.com.